Welcome everyone to this week's Journal Club. Thank you, Ken, for letting SNEV help us moderate today. And our presenter for today is Chungmin Han, who finished his PhD in August last year at the Department of Mechanical Engineering in Pohang University of Science and Technology. Now he's working as a postdoctoral researcher at the School of Interdisciplinary Biosciences and Bioengineering in the same university. The paper that he's presenting today is his PhD dissertation work, and it's about the tetraspanin analysis of individual EVs using total internal reflection fluorescence microscope, using total reflection fluorescence microscope. So Chungmin, feel free to start.
Thank you, Camille, for the kind introduction. Hello, everyone. I'm Chungmin from PostTech, Pohang University of Science and Technology. It's in South Korea. It's a great honor for me to introduce my recent work to the international EV researchers today. For a brief self-introduction, I received PhD last year, as Camille introduced, and I'm currently working as a postdoctoral researcher at Jae-sung Park's laboratory at PostTech. My research is mostly focused on single-vesicle analysis and its applications. And for today's presentation, I'm going to talk about details of single-vesicle analysis methods that our laboratory have developed. And then I will talk about result of HEC-293 cell single EV analysis. I first presented basic principles of this work at the ISCB 2018. And the work finally came out as a paper at the JEV in this year. So if you like the presentation today, please search and read this paper too. I think I don't need to talk much about basic concepts of EVs because most of the listeners of today are experts of EVs. So I'm going to briefly mention about a few key takeaway messages from the slides. So the important message that I would like to talk in this slide is that first, EVs consist of various different subpopulations. For example, exosomes and microvesicles, or ectosomes, exomeres, and etc. Second, because of these various subpopulations, a sample of EVs are expected to be very heterogeneous. And because of these heterogeneous characteristics of extracellular vesicles, analyzing or reducing its heterogeneity became one of the most important parts of investigation of extracellular vesicle investigation today. And EVs are not only heterogeneous, but also very complex. As far as we know, extracellular vesicles are composed of hundreds of different biomolecules, including proteins, lipids, and nucleic acids. And thanks to the recent omics approaches, today we have a very comprehensive list of proteins, lipids, and nucleic acids that make up extracellular vesicles. Based on the omics result, we selected a number of hallmarks protein of EVs, such as CD9, 63, 81, and we have been using these proteins in our investigation as EV identification markers. Although the omics approaches revealed very important information of extracellular vesicles, they have a significant limitation that they can only provide averaged information of heterogeneous extracellular vesicle population. So to visually demonstrate this limitation, I drew a simple example in this slide. Here I drew two different EV samples. Sample 1 has 3 different EVs, and sample 2 has 3 identical EVs. When these 2 different samples were analyzed by omics approach, the result will show these 2 samples are same, and which is not true. This kind of error is made when EVs in the sample were licensed to be used for the ensemble averaging method, like omics or Western blots, and etc. So therefore, to understand true characteristics of EV samples, we should utilize techniques that can analyze characteristics of a single extracellular vesicle particles. Actually, there are some single vesicle analysis techniques that are actively used in current EV researches. Some of them are very essential to characterize isolated EVs. For example, If we don't perform electron microscopy or NTA analysis, we probably cannot publish our research in any journals. So some of them are very essential nowadays. However, these widely used single-vesicle analysis techniques can only analyze these physical properties of extracellular vesicles. For example, they can provide size and morphology and surface charge of single extracellular vesicles, but they cannot provide any biological or chemical properties of single extracellular vesicle particles, such as their protein expressions. Therefore, these techniques actually cannot solve the limitations of conventional ensemble averaging analysis that I just mentioned in the previous slide. So to overcome these technical limitations of current single EV techniques, a few number of new single vesicle analysis devices have been recently proposed. Most of the techniques are using fluorescent labeling for analyzing biological characteristics of single extracellular vesicles. Although these specialized devices can provide a new and promising single EV analysis method, the costs are too expensive to be purchased by individual research groups. Therefore, the number of research articles using these devices are not sufficient yet to evaluate the reliability of their analysis. So in this current situation, I actually had an old question that started from early period of my graduate study. So my question was, whenever I performed Western blot analysis of tetraspanin markers, I was always curious about how these tetraspanin markers were actually expressed in single vesicles. Are they all expressed in one EV particle, or are they expressed one by one in different vesicle, or are there any specific expression patterns? So no one could answer this question because there were no available single vesicle analysis method in the field of extracellular vesicle at that time. So to answer this question, I decided to build a new single vesicle analysis that can be performed using an affordable conventional laboratory equipment. First of all, I decided to use total internal reflection microscopy to build a new single vesicle analysis. There were two major reasons for choosing turf microscope. First, turf microscopes are relatively cheap and easy to find in nearby laboratories. And second, a turf microscopy produces excellent signal-to-noise ratio images that can even detect a single fluorescent protein. Also, because a turf microscope only visualizes objects located within 200 nanometer distance from the reflection surface, it is also very useful to visualize EVs because EVs also have an average size of 100 to 200 nanometers. And in addition to the basic turf microscope system, to analyze expression of multiple markers in a single vesicle, I made some modification in conventional turf microscope system. First, I added 4 aligned excitation lasers for multicolor simultaneous fluorescent excitation. And second, I added commercially available 4-color simultaneous imaging optics system that can spatially split 4 different fluorescent emissions into one CCD camera. So after all microscopy systems was established, the multicolor imaging capability of the system was tested using EV-sized multifluorescent nanoparticles. The results showed that all 4 fluorescent signals from the EV-sized nanoparticles were successfully observed as well-isolated individual spots in our system. Before moving on to the experiment using real EV samples, we performed an additional demonstration of co-localization analysis using 2 different EV-sized bead samples. So one sample was 100 nanometer fluorescent multicolor fluorescent beads that every individual particles having both the red and green dyes. And the other sample was 100 nanometer red and green bead mixture. So if we analyze these 2 samples using ensemble averaging measurements, such as plate leader, we could not detect the difference between the samples. However, the images acquired from our microscopy system successfully demonstrated the difference between the samples, and further co-localization analysis of the images quantitatively showed this difference very successfully. And since all demonstration using EV-sized nanoparticles were very successful, we next moved on the characterization of Lear EV particles using HEC-293 cell EVs. One important thing is, unlike fluorescent nanoparticles, EVs do not generally contain endogenous fluorescent molecules. And therefore, we need to label them before the analysis. So most commonly used EV labeling and washing methods are ultracentrifugation and side exclusion chromatography. However, those methods are generally time-consuming and requires large amount of EVs. Therefore, we should find another way to solve this problem. To solve this problem, we try to adopt a twin passivated glass surface for efficient EV immobilization and fluorescent labeling. Using this surface, EVs are immobilized on the surface by strong biotin-avidin interactions, while all other molecules, including fluorescent antibodies, cannot adhere to the surface because the surface is passivated by twin molecules. Thanks to these unique characteristics of the surface, we can perform entire process of single EV analysis, including EV immobilization and fluorescent labeling, only within an hour. So to visually demonstrate the immobilization of a biotinylated extracellular vesicles on the twin surface, we used EVs isolated from HEC-293 cell expressing palmitoylated GFP. When we introduced the biotinylated extracellular vesicles to the surface, EVs were well immobilized on the surface, as seen at this video clip. On the other hand, when EVs without biotin were introduced to the surface, they cannot attach to the surface and kept diffusing. When 20 frames of each videos were averaged to produce noise-reduced images, only the biotinylated and immobilized EV sample showed bright signals from single extracellular vesicles. And we double-checked the EV immobilization using two different electron microscopes. One is scanning electron microscopy and the other is a scanning probe microscope. From the result of both microscopes, we could confirm that only the biotinylated EVs were immobilized on the surface, and the size of EVs detected from the both analysis were well corresponded with the previous literatures. And then we tested antibody repelling ability of the twin passivated surface. As you can see on the images on the left side, twin passivated surface dramatically reduced non-specific binding of fluorescence antibodies compared to the control glasses. So next, we optimized the antibody labeling condition using HEC-293 EVs, which is isolated using differential ultracentrifugation. From the result, we confirmed that the number of detected EV signals were saturated at 3.3 microgram per milliliter antibody concentration in all three tetraspanin antibodies. And the surface seemed effectively block non-specific bindings of antibodies up to 100 microgram per milliliter. So based on this characterization, our following tetraspanin labeling analysis were performed with at least 10 microgram per milliliter antibody concentration. And finally, we applied this turf-based single-vesicle analysis method to investigate tetraspanin expression profile of real EV samples. So EVs were isolated from HEC-293 cells by differential ultracentrifugation. The images on the left side are representative turf microscopy image of HEC-293 EVs with tetraspanin labelings. From the magnified images, we can confirm that there are three different types of extracellular vesicles. The first one is CD96381 triple positive EVs. A second one is a CD63 single positive EVs. and the other is CD981 double positive EVs. To more quantitatively analyze this tetraspanin expression profile, we analyzed more than 5,000 single EV signals from about 15 different images. And from the result, we identified 5 different populations of EVs that had different tetraspanin expression profiles. About half of the EVs were tetraspanin single positive EVs, and the other half of the total EVs expressed more than 2 different types of tetraspanin at the same time. One interesting observation is that there were almost no CD963 double positive EVs and CD6381 double positive EVs. When we expressed detected population EVs in a Venn diagram, it appeared as shown in the slide. The entire populations were roughly divided into 2 groups. One was CD63 single positive EVs, which took about 34% of total EVs. And the other was CD9 and 81 double positive EVs, which again took about 34% of total extracellular vesicles. So using our turf-based single vesicle analysis, now we can answer the question how tetraspanins are expressed in single vesicles. The rough answer is that HEC29, in the case of HEC293 cell EVs, they are composed of CD9 and 81 double positive cell populations and CD63 single positive cell populations. In this situation, next reasonable question will be something like, then what are these cell populations, or are they exosomes or microvesicles? Although this is very important question, and I'm actually currently investigating on this topic, I still need to collect more experimental evidences to answer these questions. Therefore, today, instead of talking about these topics, I would like to talk more about how EV purification method affect cell populations of purified EV samples. And to investigate the effect of EV purification method, we further purified EVs using 2 different OptiPrep density gradient method and 1 side exclusion chromatography method. And we collected all fractions from the purification method and analyzed their EV cell populations using our single vesicle analysis method. So all of the collected fractions were analyzed using the single tetraspanin analysis, a single vesicle tetraspanin analysis. Images on the slides are representative turf image of the all fractions. And this suggested that density gradient 10 to 20% fraction isolated largest amount of tetraspanin expressing extracellular vesicles. On the other hand, buoyant density gradient 0 to 10% fraction were the lowest yield fraction that showed too low number of signals to be quantified for the further population analysis. When EV subpopulations of density gradient fractions were further analyzed using tetraspanin co-localization, we could figure out that the EV populations of 10 to 20% fraction were completely different from the EV subpopulations of 0 to 10 or 20 to 30% fractions. Most distinct difference was the CD63 single positive population. More than half of the EVs from density gradient 0 to 10 and 20 to 30 fractions were CD63 single positive. The density gradient 10 to 20 fraction only had less than 5% of them. On the other hand, while almost half of the EVs from the density gradient to 10 to 20 fractions were CD9 and 81 double positive, less than 20% of EVs from density gradient to 0 to 10 and 20 to 30 fractions were CD9 and 81 double positive. So comparing this result of density gradient fractions with the differential ultracentrifuge EV results, we could conclude that density gradient 10 to 20 fraction purified CD9 81 double positive EVs from these DUC EVs. And the density gradient 0 to 10 and 20 to 30 fraction purify CD63 single positive fractions from the DUC EV samples. The co-localization analysis result of buoyant density gradient fractions were very similar to that of density gradient fractions. Again, almost half of EVs from 20 to 30 fraction were CD63 single positive, but buoyant density gradient 10 to 20 fraction only had less than 5% of them. And more than half of EVs from buoyant density gradient, in the case of buoyant density gradient 0 to 10 fraction, the number of detected tetraspanin signal was too small, so the result of analysis seemed a little bit unreliable. Again, when comparing the result of buoyant density gradient fractions with differential ultracentrifuge EV result, we could conclude that buoyant density gradient 10 to 20 fraction purify CD981 double positive EV populations. And the buoyant density gradient 20 to 30 fraction purifies CD63 single positive fractions from the DUC EV samples. The only distinct difference between buoyant density gradient and density gradient fractions was their yield. Density gradient purification generally isolated more tetraspanin expressing EVs than the buoyant density gradient method. Finally, unlike the previous result of buoyant density gradient and density gradient fractions, SEC isolated fractions did not show much difference in terms of their EV subpopulations. SEC 11 to 12 and SEC 13 to 14 fractions showed almost a similar subpopulation with the original differential ultracentrifuge EVs, and SEC 15 to 16 fraction showed slightly more CD9 and 81 double positive EVs, but it still includes significant amount of CD63 single positive extracellular vesicles. And therefore, based on this observation, we could confirm that there was no significant correlations between tetraspanin expressions and the size of extracellular vesicles. And when the result of the all fractions were collected and plotted in one TSNE plot, we can observe 4 distinct clusters from the analysis. From the cluster 3 and 5, we could confirm again that density gradient and buoyant density gradient fractions purified similar EV subpopulations. And from the cluster 4, we could reconfirm that the EVs in different SEC fractions did not show much difference in terms of tetraspanin expression profile. And so this is the last slide of the presentation. In summary, turf-based single-vesicle analysis successfully revealed subpopulations in HEC293 cell EVs. According to our result, the sample prepared using differential ultracentrifugation had 2 different major EV subpopulations. One is CD981 double positive EVs, and the other is CD63 single positive EVs. And from the further single-vesicle analysis using EVs acquired from different purification methods, we could confirm that these 2 major subpopulations can be further purified. So density gradient 10 to 20, buoyant density gradient 10 to 20, and SEC 15 to 16 fractions mostly isolated CD9 and 81 double positive EVs, and the other fractions of purification method mostly isolated CD63 single positive EVs. So this is the last slide. Thank you for listening, and if you have any questions, I will answer those.
Thank you, Chungmin, for that presentation. We'll move on to the questions. So I see several questions here in the chat box. I'm gonna call on Artem first.
Yeah, hi, thank you. It's an amazing presentation, amazing technique. And this is the way how we can study vesicles in the level of single EVs. But as I know, there is several other equipment which is commercially available. For example, ExaView, which which is based on a very similar way. So my question is, have you ever compared your data and, for example, some commercially available equipment?
Your question was like, did I compare my research with any other commercially available single vesicle analysis? Is that right?
Yes, that was correct. Yeah, you heard that correctly.
So I actually had, I didn't have any chances to access these single vesicle, commercially available single vesicle analysis devices. So I actually don't have any result of comparison, but I recently read some papers, bioarchive papers actually, using those devices to analyzing tetraspanin expressions of single extracellular vesicles. And what I remember is their results were almost similar with my turf-based extracellular vesicle analysis result. So I think maybe the results are comparable, and yeah, that's my answer.
Okay. Our next question is from Alyssa.
Hi, thank you very much for your talk. I wanted to know, you had mentioned the tetraspanin proteins. Do you know if there were any different tetraspanin proteins on low versus high density EVs? Thank you.
Can you repeat the question again? Sorry.
Sure. So do you know if there are different tetraspanin proteins on low versus high density EVs?
Oh, so in terms of density gradient, I analyzed low and I have analyzed low and high density extracellular vesicles because I analyzed the EVs from density gradient 0 to 10 and 20 to 30. But these two low and high density EVs actually expressed very similar patterns of tetraspanin. The only difference is density gradient 10 to 20 fraction, which is just medium density extracellular vesicles, which have distinctly different tetraspanin expression patterns. So I actually have no idea why these low and high density extracellular vesicles expressed same sets of tetraspanins, but that's my observation.
Okay, so the only difference you saw was really in the medium density between 10 and 20.
Yes.
Perfect, thank you.
Thank you.
And then we have Leia.
Yes, so thank you for your presentation. It was really nice.
Thank you.
So my question was in the graph, it's you show the total EVs. So do you have a general marker that you're using, or it's just the sum of all the EVs you see?
Oh, okay. So I actually tried to measure the total EVs during this project, but I failed to find the general marker for total EVs. I actually tried various different lipid labeling techniques, including DI dyes and cell masks, and also cholera toxins, and something like all those lipid markers I have tried, but those are actually not label all of the extracellular vesicles. They only labeled a small portion of extracellular vesicles. So I actually failed to label and measure the total amount of EVs. Yeah, that's the answer. So I tried, but I think there are no such markers to label all the EVs in the media or in the sample.
Okay, thank you. Because I am using Membrite in flow cytometry. I'm also trying to analyze single vesicle. So I'm curious.
Thank you.
So for the next question, we have Meline.
Yeah, sorry. Can you explain how the biotinylation of the vesicles, of the biotin vesicles works?
Okay, so to biotinylate extracellular vesicles, I used a product called NHS biotin, which is conjugating biotin to the amines, primary amines of proteins on the extracellular vesicles. So to use this NHS biotin, I first pellet the extracellular vesicles using ultracentrifugation and resuspend them in a small amount of buffer and conjugate it with NHS biotin. So NHS biotin and wash them again using differential ultracentrifugation. So that's the process I used, and yep, that's it.
Okay. The next question is from Se Kyung Tae. I'm sorry if I'm butchering your name.
Yes. Thank you for the talk.
Thank you.
Yeah, I just want to ask a few questions. As mentioned in your M&M parts in your publication, the EVs were collected from cells culture in supplement-free based media. So any reason for not adding the EV depleted FBS as the supplement in the media?
We sometimes use EV depleted FBS, but in my opinion, it's almost impossible to get rid of all exogens from FBS. So there is always a possibility that FBS derived EVs are contaminating our samples. So we did not use this EV depleted FBS. And also in the case of HEC293 cell, culture these cells in serum-free based media for 24 hours actually did not affect the cell viability that much. So instead of using EV depleted FBS, we just used a supplement-free based media.
Okay, thank you. So do you observe any cell death after 24 hours?
Sorry, can you ask again?
Just now you mentioned, sorry. Yeah, just now you mentioned that the cell you did not observe any cell death in the 24 hours incubation times of the HEC293 cells, right?
Yes, yes.
Yeah, so how about is like after 24 hours, like 48 or 72 hours of incubation? So do you ever try to incubate such one?
Yes, I tried, and cells were shrinked a lot, but they didn't look, they didn't seem like they are dying, but they shrinked a lot, and it seems like it's open over. So I just used only for 24 hours.
No, okay. Another question, so a total of 6 liters of conditioned medium was pooled for EV collection. So are these conditioned medium collected from the same generation of cells?
Oh, yes. We
okay.
cultured a lot of cells at the same time and collected the media and pooled them and storage them and processed them at the same time.
Okay.
To reduce the variations. Okay.
Okay, just one more question, sorry. So why you choose the DDS twin 20 surface coating for the EV immobilization? So do you ever compare with other types of coating?
I actually used a pegylated surface first, but pegylated surface also provide a very good passivation, but preparation process is a lot longer, and the cost for preparation is also more expensive in the case of pegylated surface. So, and also the storage is also a problem for pegylated surface. So we decided to use DDS surface instead of using pegylated surface.
Okay, thank you very much.
Thank you.
So we have a question from Lilia.
Yeah, thank you so much. It just like very technical question, but because I just faced the problem with the MATLAB when I analyzed like the blue and the green intensities from the different colors for the vesicles. I'm really wondering, how did you solve the problem when the two EVs, let's say, very close to each other overlap, and then you have an overlapping signal from CD81 and CD9? Because from my experience, it was like too much noise and the error was very high, and so I was just going for MATLAB instead of that, just using the area scanning. So I was a little bit surprised, and if you got maybe some more information somewhere in your paper's reference, it will be really appreciate, because I'm afraid that the data about the CD9 and CD81 double positive EVs may be a little bit too far away from the reality. So what do you think?
Oh, actually, I didn't use blue color for labeling CD81. I actually used yellow color, 561 nanometer laser for CD81. And that's one thing. And also there should be an overlap over emission spectrum. But if you take a control image of only one laser excitation and measure that overlapped intensity of, what can I say, bleed through intensity of other color, bleed through intensities from other color channel. If you measure this intensity from the control images, and you can subtract this intensity when you process your image, and then you can reduce the risk of misreading the co-localization result from your processed image. Did I answer correctly, or do you understand what I just said?
Yeah, I'll just go, I think, to the methods in the paper. Yeah, I got it. Thank you.
Thank you.
Then we have Benedetta.
Oh yeah, thank you. Very nice presentation, really. I have two questions. One is related to the specificity of the EV type. So did you have the chance to test another cell type? And the other, I will finish, is do you think that your different expression could also depend on the affinity of different tetraspanin antibodies? So one could be better than another?
Mm-hmm. So for the first question, I have tested like more than 7 different cell types. So I'm actually currently working on this topic. When I analyze extracellular vesicles from the epithelial cells, they have very similar tetraspanin expression patterns. But if I collected extracellular vesicles from other cell types like melanoma or mesenchymal stem cells, they have very different tetraspanin expression patterns. So I think, yeah. So that is the first answer for the first question. And the second question was about the antibody, right? Yeah. To select antibody, we tested at least 4 different clones of antibodies for each tetraspanins, and we selected two antibodies that gave us most strong signals, and compare the count of the signals from analysis. And these 2 antibodies were almost comparable. So we selected one of those antibodies, and some of the antibodies have a very limited choices for their fluorescent color selections. So in that case, we alternatively use these 2 different clones.
Yeah, okay, very precise, thank you. Yeah, we have some data comparing different instruments, and we really figure out that different expression could be due to different antibodies used.
Ken has a follow-up question for Ken.
Yeah, I think that Benedetta's question is really important. So I mean, not only the different cell lines and the different antibodies, what the results may be, but also what about growth conditions? So if you have cells that are in different passage numbers or at different densities of culture, even different supplements in the medium. I mean, I wonder if you can speculate on whether you think that might also affect how many, I mean, we know it affects how many vesicles are released, the density, for example, but it could also be that it would affect tetraspanin expression. Do you think that that's plausible?
So the question was a growth condition, and what was the other thing?
Yeah, so I'm just wondering if you think it is plausible that the growth conditions, including cell density, including maybe the the even a matrix that the cells would be grown on, that any of these things could also affect our our ability to compare what happens in my lab to what's happening in your lab?
So I didn't actually test it that details of growth conditions. So I always seed the cells in the same density, and I always culture cells in the same media. But what I actually tested is I tested the EV depleted FBS condition and serum free condition of HEC293 cell culture. And these two different conditions actually gave me almost a similar tetraspanin expression patterns. But the growth condition actually gave me a different yield of extracellular vesicles. So that's one thing. And the passage, I actually didn't test about, actually test the different passage cells, because the cells I cultured are cancer cells, so we didn't actually track the passage of the cancer cells. So I cannot answer the question.
Thank you for that. Yeah, I think it's important because different labs now are starting to do some inter-lab comparisons. And at one level, I guess we can share the same material, but I think as we start to try to understand biological differences, we're going to be also culturing the same cells in different labs and seeing if we get the same results. And I guess it's important to keep in mind that there is the possibility for some of those variabilities. So thanks for the answer.
Thank you.
So the next two questions we have are kind of in a similar, or they're kind of similar, so I'm just gonna ask them. So one is from Martin Wolf and the other is from Kyung-hyung Lee. And their question is, how sure are you that what you're seeing is in each signal or each spot is a single vesicle and not an aggregate? So I have a similar question in mind earlier. Are you getting any of that swarm? Like, is there a chance that if you're incubating too much EVs, are you getting this swarming, or are you seeing that you're not seeing just one particle, but multiple particles that are aggregated?
So I actually asked the question when I submitted my paper to journal, and the experiment I performed to answer the question was I added a certain amount of detergent to the EV samples to dissociate aggregates. And when I add a small amount of detergent, it did not change any result. But when I add when I add more amount of detergent, then the signals were just disappeared because EVs were disrupted by detergent. So that was the one evidence I submitted to the reviewers. And another thing that I performed is we actually filtered after centrifugation and biotinylation to remove the aggregates of extracellular vesicles. And it also did not change the overall profile of tetraspanin expressions. So from those results, we think our sample has very low amounts of aggregated extracellular vesicles, or almost no aggregates. That's what we think now. And it is really hard to tell that single spot is actually a single extracellular vesicle or a tight aggregate of small extracellular vesicle. It's really hard to tell. So we can only provide indirect evidences to answer the questions.
So I have some more questions for you. I mean, what is the minimum particle size range that you're detecting using this method, and how long does the whole process take from preparing the slide to imaging? Because you mentioned biotinylation takes an hour, but like from preparing the slide until imaging and counting the particles, how long does it typically take?
So preparing the slides actually take about 4 to 6 hours, but I can handle about 20s of glass slides at the same time. So that's one thing. And for analyzing vesicles, I should assemble these glasses into small flow chamber, and it took about an hour to cure the resins for making flow chamber. And the other part is making surface functionalized and put EVs immobilized and EV labeling, and those all process take about an hour. So that's one thing. And what was the another question? Sorry.
So yeah, that I mean, so in terms of like counting the particles and how long does that take? So it's around 8 hours now, like so 6 hours for 4 to 6 hours preparing the slide, and then an hour for incubating the EVs, and then image analysis. How long does that usually take you per slide? I'm just curious with like how long the whole process goes.
Okay. Did I answer the question?
Yeah, just in the imaging part, how long does it take you to analyze a slide then?
Oh, image process, you mean? Oh, so taking images take about 10 minutes, and processing the images actually, the image process were actually performed using computer code. So it took about 10 minutes or so. So it's not that much.
And then I think the other question I had was, what's the minimum particle size range that you're detecting using this method?
Oh, okay. So I'm using fluorescent imaging, so I cannot actually measure the size of extracellular vesicles. So I actually don't know, but the turf microscopy actually can detect a single fluorescent protein, like a single GFP also can be detected by turf microscopy and CCD setups. So I don't know the size of extracellular vesicles, but it can detect a single fluorescent molecule. So that's my answer.
Thank you for that. We also have one more question from Jake Yong-tae.
Yes, sorry, just one last question. So do you think your immobilization method applicable to super resolution imaging?
So what type of super resolution imaging are you planning to use?
I think I'm looking at the device calling the nano bioimaging. So using the STORMS method.
So immobilization, there are no other reason the immobilization didn't work for super resolution imaging. So I think you can use it for storm imaging. Actually, in the case of palm super resolution imaging, many people using this kind of passivated surface. And yeah, there are no reasons that this surface did not work for super resolution imaging.
Okay, thank you very much.
Thank you.
We have no more questions in the chat, but I have one last question, I guess. In terms of incubating EVs on the slides, are you doing incubation by count or just by volume? Are you doing NTA before putting these EVs on the slide?
I tested all conditions. I incubated EVs by counts, by protein amounts, and by volumes, and the result was not so different between these three different methods, because the capability of the surface, it has limited space for EV immobilization. So if I put enough amounts of EVs, the saturated counts of EVs are not that different between the different EV incubation criteria. For example, protein amounts, counts, and volumes, they all produce similar amounts of EV counts.
Okay, thank you. We have no more questions in the chat. So again, I want to thank Chungmin for his presentation, and also Ken, thanks again for letting SNEV co-host this journal club. Thanks everyone for your questions, and thank you for joining us today.
Thank you, Camille.